Hello friends, today we are going to discussing about what are the difference between prokaryotes and eukaryotes. You all know prokaryotes are organisms which doesn't contain nucleus, whereas eukaryotes are organisms which contain nucleus. So what are the main difference between prokaryotes and eukaryotes? We are going to discuss in this class. So if we take the ultrastructure of prokaryotes and eukaryotic cells as shown in the diagram. So it is prokaryotic organism, that is bacteria. So all prokaryotes are called bacteria. So bacteria is a prokaryotic organism. There's other type of organisms like Protista, fungi, plants, animals. All these are eukaryotes. If we take a five kingdom classification, except Monera, remaining all living organisms are considered as eukaryotes. Eukaryotes. For example, Monera means prokaryotes, all bacteria. So all bacteria called Monera and all these Monera is known as prokaryotic organism. Whereas other types of organisms like Protista, fungi, animals, plants, all these are considered as eukaryotes. Eukaryotes mean they contain prominent nucleus in its cell, whereas prokaryotes doesn't contain any nucleus. That means these are single membrane bound organisms, whereas eukaryotes are multi-membrane bound organisms. So if we take prokaryote ultrastructure, it contains different parts. The outermost part is capsule. This capsule is mostly made up with carbohydrates and it is helpful for the prevention of desiccation or dehydration. Sometimes it also helpful for the attachment to the substrate. So this is the outermost layer of a prokaryotic organism or outermost structure of a prokaryotic organism. Beneath this, cell wall is present. Mostly, the cell wall is made up with peptidoglycan. You all know peptidoglycan is a polymer of murin, and murin is a polymer of a two component unit, that is NAG and NAM. So cell wall is made up with bacterial cell wall is made up with peptidoglycan, and below this cytoplasmic membrane is present or plasma membrane is present. Plasma membrane is a dynamic structure which involved in the membrane transport and also involved in different types of exocytosis and endocytosis function. This is the membrane. Below this, cytosol is present. Why it is called cytosol means the liquid portion of the prokaryotic cell doesn't contain any membrane bound organelle. That's why it is called cytosol. Within the cytosol, ribosomes is present. These ribosomes are protein synthesizing machineries. Generally, in prokaryotes, 70s ribosomes are present. These 70s ribosomes are protein synthesizing factories, are protein synthesizing missionaries. Attached to the capsule, there is a small thread-like structures are called there. These thread-like structures are called pili. These pili mostly involved in conjugation. During conjugation, there is a conjugation tube formation taking place. That conjugation tube made with these pili. Pili involved in gene transfer during conjugation. And this is cytosol. The liquid portion of the bacteria is known as cytosol, not cytoplasm. And this blue color structure is called DNA. This is called nucleoid because there is no true nucleus is present. It is distributed along the cytosol. That's why it is called nucleoid. This is DNA portion. Another name for this nucleoid or DNA is known as genophore. So either nucleoid or genophore, we can use the terminology for bacterial DNA. Some bacteria contain closed circular DNA molecules called plasmids, and these plasmids involved in defense mechanisms. And while conjugation time also, these plasmid DNA only transferred. It replicated and transferred. The small circular double-stranded DNA molecule present in some bacteria are called plasmids, and these plasmids mostly involved in defense mechanisms. But this major DNA, that is nucleide or xenophore, involved in general biological functions of the bacteria. And another organelle that is present in called flagella. This flagella involved in the movement of the bacteria, locomotion of the bacteria. So these are the different parts of bacterial structure, ultrastructure of bacteria. So all bacteria comes under prokaryotes. If you take eukaryotes are different types of eukaryotes are present. Those are protists, fungi, animals, and plants. All these are eukaryotes. In these eukaryotes, except animals, remaining all cells contain cell wall. For example, if you take Protista, it also contains cell wall. Fungi, it also contains cell wall. Plants also contain cell wall. But animals doesn't possess any cell wall. Even though the ultrastructure of the eukaryotic cell has some common components. To take the structure, this is microtubules. So microtubules are involved in the movement of chromosomes while cell division. So these microtubules are involved in movement of chromosomes while during cell division. They form spindle fibers. And second one is mitochondria. These are the powerhouses of living cell. They are involved in uh, carbohydrate metabolism, lipid metabolism, and also ATP synthesis also it is involved. That's why it is called powerhouses of living cell. Do you know this mitochondria? Once upon a time, it is a bacteria. During evolution, it entered into eukaryotic cell and settled in the eukaryotic cell. So this mitochondria is a bacterial origin. According to endosymbiotic theory, mitochondria is a bacterial origin. Next, centriole. The centrioles present mostly in animal cells. And the centrioles are responsible for the production of microtubules. So these microtubules are produced from centrioles. Centrioles exclusively present in animal cells. And these are the vesicles. Vesicles means they are exporting or importing mechanism. Whatever the waste material is present, that waste material is exported out from the cell by these vesicles. Or whatever the important things are coming to the cell, they are also through vesicles. These vesicles are formed by Golgi complex. And this is cytoplasm. In eukaryotic cells, the liquid portion of a cell is known as cytoplasm because cytoplasm contains liquid portion plus membrane bound organelles. So membrane bound organelles plus liquid portion commonly called as cytoplasm. It is not cytosol, it is a cytoplasm. Bacterial liquid portion is called cytosol, whereas eukaryotic liquid portion is known as cytoplasm because they contain plasma membrane bound organelles along with the liquid portion. Then rough endoplasmic reticulum. This rough endoplasmic reticulum means endoplasmic reticulum with the ribosomes. Whenever ribosomes attach to the endoplasmic reticulum, then it is called rough endoplasmic reticulum. And endoplasmic reticulum connects nucleus with the plasma membrane. So what is the connection between nucleus and plasma membrane means endoplasmic reticulum. If this endoplasmic reticulum attached to the ribosomes, then it is called rough endoplasmic reticulum, generally involved in protein synthesis. Another one is smooth endoplasmic reticulum. If endoplasmic reticulum not attached to the ribosome, then it is called smooth endoplasmic reticulum. Smooth endoplasmic reticulum involved in the lipid biosynthesis. Then ribosomes. Generally in eukaryotes, cytosol contain 80s ribosomes, whereas mitochondria contain 70s ribosomes. As I said earlier, mitochondria is a bacterial origin. That's why mitochondria contains 70s ribosomes, whereas other portion of the eukaryotic cell contain 80s ribosomes. So these 80s ribosomes are the protein synthesizing factors. This is the plasma membrane. Eukaryotic plasma membrane is a highly dynamic structure. It is involved in export and import of the materials and also charge generation, and also it is uh, the plasma membrane also shows lateral and flip-flop movements also. That's why it is very dynamic structure. This is plasma membrane and this is flagell. Some cells of eukaryotic cells or animals have the property to move from one place to another place. So that mitochondria, that cells contain flagellum. But the flagellum of bacteria is different from eukaryotic cells. Generally, the bacterial flagella made up with flagellin protein, whereas eukaryotic flagella made up with microtubules. And this is a lysosome. These are the suicidal bags. Whenever any material or organelle damaged in the cell, that is recycled by the lysosome. That's why they are called suicidal bags. And in eukaryotic cell, highly polymerase or polymorphic organelles, highly polymorphic. That means different shapes appear. Organelles are lysosomes. So highly polymorphic organelles in eukaryotic cells are lysosomes. Then Golgi complex. Golgi complex involved in the secretion function. That means whatever the material want to go out the cell or come into the cell, these are export and import process or secretion process is taken care by Golgi complex. Then this is chromatin. Chromatin means DNA plus histone proteins is known as chromatin. DNA plus histone proteins is known as chromatin. Is chromatin further condensed into chromosomes. So generally, if DNA attached to the histone protein loosely, then it is called euchromatin and it is majorly expressed portion of the chromosome. If DNA attached to the histone protein tightly, then it is called heterochromatin. So this way, in DNA plus histone proteins collectively called as chromatin. If DNA attached to the histone proteins loosely, then it is called euchromatin. Whereas DNA attached to the histone tightly, then it is called heterochromatin. Then this nucleus is surrounded by the nuclear membrane. This is the distinct feature of eukaryotes and prokaryotes. Prokaryote doesn't have any nuclear membrane, whereas eukaryotes contain nuclear membrane or nuclear envelope. And this nuclear membrane contain nuclear pores, and these nuclear pores are involved in export and import. Whatever the material come out of the nucleus, or whatever the material come into the nucleus, these things are made by these nuclear pores. And in nucleus, there is another portion is called that is called nucleolus. Nucleus, there is another portion called nucleolus. This is called different parts of the nucleus. So let's take brief and difference between the prokaryotes and eukaryotes. Here, this is prokaryote and this is eukaryote. Prokaryotic cell size is 0.1 to 5 microns. The size of the prokaryotic cell is 0.1 to 5 microns, whereas size of the eukaryotic cell is 5 to 100 microns. So prokaryotic cell is smaller than eukaryotic cell. Then prokaryotic cell nucleus is absent, whereas eukaryotic cell nucleus is present. All eukaryotic cells contain nucleus, whereas prokaryotic cell doesn't possess any nucleus. Prokaryotes doesn't have any membrane bound organelles and also nucleus. So absence of membrane bound organelles and nucleus is seen in prokaryotes, whereas eukaryotic cells contain membrane bound nucleus and organelles also, like mitochondria, chloroplast, endoplasmic reticulum. All these things appear in eukaryotic cells. Next, one chromosome is present, but not two chromosome. That's why it is called nucleide or genopore. Only one chromosome is present that is not attached to the proteins. It is circular chromosome is present. Only one chromosome is present. In eukaryotic cell, more than one number of chromosomes present. If we take humans, there are 46 chromosomes present. Whereas in prokaryotes, only one chromosome is present. That is also not a true chromosome. Whereas in eukaryotes, several number of chromosomes present. And prokaryotes are unicellular organisms. All prokaryotes are unicellular organisms, whereas eukaryotes are mostly multicellular organisms, except the protist, remaining all eukaryotes are multicellular organisms. And lysosomes and peroxisomes are absent in prokaryotes, whereas lysosomes and peroxisomes present in eukaryotes. Microtubules absent in prokaryotes, whereas microtubules present in eukaryotes. Endoplasmic reticulum also absent in prokaryotes. Endoplasmic reticulum present in eukaryotes. Next, mitochondria is absent in prokaryotes, whereas mitochondria present in eukaryotes. Cytoskeleton absent in prokaryotes. Cytoskeleton is present in eukaryotes. Generally, cytoskeleton is the uh, internal skeletal structure that gives support to the cell. Generally, this cytoskeleton made up with three components. Those are actin, tubulin, and these things are present. Actin and tubulins. These two components are present in the cytoskeleton. So cytoskeleton supports the cell shape, structure, and internal transport and everything supported by the cytoskeleton. The cytoskeleton is present in eukaryotes but not in the prokaryotes. In the ribosomes, if you take ribosomes, prokaryotic ribosomes are 70s ribosomes. Whereas in eukaryotes, there are two types of ribosomes present in the organelles like mitochondria and chloroplast. 70s ribosomes present because they are bacterial origin. Whereas in eukaryotes, 80s ribosomes present. That means cytoplasm, 80s ribosomes present. Whereas in mitochondria and chloroplast, 70s ribosomes are present. That means both 70s and 80s ribosomes present in eukaryotes, whereas only 70s ribosomes present in prokaryotes. Vesicles present. Vesicles present. Both cases, vesicles are present because transport of materials need to both prokaryotes and eukaryotes. That's why vesicles present in both prokaryotes and eukaryotes. Golgi apparatus is absent in prokaryotes, whereas Golgi apparatus is present in eukaryotes. Chloroplast absent. Chlorophyll scattered in the cytoplasm. Chloroplast, some photosynthetic bacteria contain chlorophyll, but chloroplast is absent in prokaryotes. Whereas chloroplast present in plants. In eukaryotes, only plants contain chloroplast. Submicroscopic in size flagella is present and made up with only one fiber. Only submicroscopic flagella is present, involved in locomotion. It made up with flagellin. Whereas microscopic in size membrane bound flagella is present. It is made up with tubulin. Cell wall chemically complex. Generally, bacterial cell wall is made up with peptidoglycan, which is a polymer of urine. Each urine contains NAM and NAG. It is present in prokaryotes. Whereas cell wall in eukaryotes are different types. For example, if you take fungi, cell wall is made up with pectin, whereas plant cell wall made up with cellulose and hemicellulose. Like that, different types of eukaryotes contain different types of cell walls, whereas bacteria contain or prokaryotes contain peptidoglycan cell wall. Vacuoles are absent in prokaryotes, whereas vacuoles present in eukaryotes. In prokaryotes, the permeability of the nuclear membrane is not present. There is no nucleus, and the nuclear membrane is also present. That's why there is no permeability. There is no difference of cytosol and nucleus in prokaryote. That's why there is no permeability. Whereas in eukaryotes, there is a clear-cut difference between cytoplasm and nucleus. There is nuclear membrane is present, and it contains nuclear pores. Nuclear pores involved in nuclear transport in eukaryotes. Sexual reproduction is absent. Generally, in bacteria, as it is single cell organism, so generally binary fission is taking place for reproduction. This is a sexual reproduction method. Whereas if you take eukaryotes, there are both sexual reproduction is present. In some cases, asexual reproduction also present, like fungi, some plants. Sexual reproduction is present, and also sexual reproduction is also present. But in case of bacteria, only asexual reproduction is present. Endocytes and exocytes are absent in bacteria or prokaryotes, whereas endocytes and exocytes present in eukaryotes. We take bacteria, it may have villi and fimbriae. It has villi and fimbriae, and these villi involved in the conjugation process, whereas in eukaryotes, villi and fimbriae are absent. And the extraordinary feature of prokaryotes is transcription occur in the cytoplasm. Both transcription and translation occur in the cytosol because there is no nucleus. That's why the both transcription and translation occur in the nucleus, and these two processes occur in simultaneously. Transcription and translation occur simultaneously in prokaryotes, whereas in eukaryotes, transcription occur in the nucleus, and these produced RNA enter into the cytoplasm through nuclear pores, and translation taking place in the cytoplasm. So there is spatial and temporal difference between the transcription and translation in eukaryotes. If you take examples of prokaryotes, bacteria and archaea. Bacteria is a modern bacteria. Archaea is a extremophiles. That means they are living in extreme condition. If you take the eukaryotes example, protists, that means unicellular eukaryotes. Protists are unicellular eukaryotes. Fungi, saprophytic organism. Plants, autotrophs are photosynthetic organism. Animals, heterotrophic organism. These are the some differences between prokaryotes and eukaryotes. Thank you.